Hi, my name is Abigail Park, and I am a senior at Carnegie Mellon University studying chemistry with a minor in computer science. I am an Amgen Scholar this summer, working with Bill Goddard and Soo Kyung Kim. And today I'll be talking about my project, Structure-Based Drug Design for TAS2R40. So just a quick overview of my presentation. First, I'll be talking about the significance of TAS2R40. Then I'll be talking about predicting TAS2R40 structure and its binding site, and then how I validated results with molecular dynamics. And finally, I'll be talking about the next steps in my project after this summer. And so bitter taste receptors are responsible for bitter taste detection, but they've also been found all throughout the body. However, their exact function is unknown, and they've actually been associated with a variety of diseases. For example, TAS2R40 has been associated with the loss of taste and also inflammatory lung disease. So the problem with studying bitter taste receptors is that there are no crystal structures available for any of the 25 bitter taste receptors that humans express. And this also makes it difficult to develop selective ligands as new therapeutic targets. So my project for the summer was to predict the structure of TAS2R40 and its binding site using computational methods with the end goal of designing a selective ligand. So next I'll be talking about the process of predicting TAS2R40 structure. So bitter taste receptors are a part of this really large family of proteins called G protein coupled receptors, or GPCRs. And GPCRs are receptors that sit in the membrane of a cell, and they are all composed of 7 helices that go through the membrane. So in this diagram, you can see here, these are the 7 helices that go through the membrane. And this is the part of the protein that's outside the cell, and this is part of the protein that's inside the cell. And you kind of see in pink where a ligand would come in and bind. And so GPCRs are humans express over 800 GPCRs, but there are less than 50 crystal structures available. Further, bitter taste receptors have very low homology with other GPCRs. And any mutations in the residues can dramatically change the structure of the protein. And this makes it particularly difficult to use homology to available structures for accurate predictions. So to mediate these obstacles, we use the GEnSeMBLE complete sampling method to predict the structure of TAS2R40. So what this entails is first we generate an initial structure of TAS2R47 helices that go through the membrane based on a related protein with known structure. And so this kind of just gives us the initial position of all the helices. And then we rotate each helix by 15-degree increments from -60 to 60 degrees. And from this step, we generate around 5 million conformations just by trying out all the different combinations of helix rotations. And then out of the 7 helices, there are 12 pairs of helix interactions. For example, helix 1 really only interacts with helix 2 and helix 7 and never really interacts with, let's say, helix 5. So we consider each of the helix pairs separately. We optimize the side chain orientations and calculate the energy for the interactions. And then we select the best 2,000 conformations from by helix. We, taking all the 12 pairs, we build the 7 helix bundles and recalculate energy. And then we select the best structures based on energy. And these structures then proceed to super bihelix. So super bihelix is similar to the bihelix method, but we're also modifying the polar angle here, which is kind of like the tilt of the helix, the azimuthal angle, which is kind of like how it rotates this way. And then we're also again rotating each helix again. And so all in all, this generates around 13 trillion conformations. So we again consider the 12 pairs, calculate the energy for those 12 pairs, select the best 2,000 conformations from super bihelix, and we build the 7 helix bundles and we recalculate energy. Then we select the best structures based on energy. So for predicting TAS2R40 structure specifically, we used the structures of TAS2R4, 5, and 14, which were determined previously using the GEnSeMBLE complete sampling method. And so in this table, you can see the identity and similarity against TAS2R40, and TAS2R4 and 5 have higher identity and similarity with TAS2R40, But even though TAS2R14 had lower homology, we went ahead and used all three of these structures as templates just so we could generate as many conformations as possible. And so then after we completed the GEnSeMBLE procedure, the structures were ranked by neutral interhelical energy, and we chose the structures that showed the most number of contacts commonly seen in active GPCR structures. And these structures were chosen to proceed with docking. And so the major difference in these conformations was seen in helix 5. So as you can see in most of the conformations, helix 5 is rotated at a negative 90-degree angle, but in SB3, it's rotated at a 15-degree angle. And this really changes the residues that are in the binding region. For example, in SB3, there's a tyrosine on the 5th helix that's pointed towards the binding site. But in SB10, that same tyrosine is pointed away from the binding region, and instead there's a valine, asparagine, and a phenylalanine pointed towards the binding site. And so now that I have the structure of TAS2R40, now I'll be moving on to how we predicted its binding site. So the method we use is called the DarwinDock complete sampling method. And so first we take all the bulky hydrophobic residues and we replace those with alanine, so we have more room for the ligand in the binding site. And then we generate a ton of different poses, around 50,000 in the binding region. And then we select the best 100 poses based on energy analysis. Then the bulky hydrophobic residues that we took out in the first step are put back into the protein, and the side chain orientations are optimized. Then we calculate two main types of energy. We call the unified cavity energy, which is the interactions between the ligand and the union of all residues involved in binding. And then we also calculate snap binding energy, which is just all the interactions between the protein and the ligand. And so TAS2R40 has 13 known ligands according to the bitter database. And out of these 13, I chose the following 5 ligands based on structural diversity, how rigid the molecule was, and how low the effective concentration was. And so as you can see, cohumulone, adhumulone, and humulone all have very similar structures, with the only difference being in this alkyl chain right here attached to the carboxyl group here. And all of these compounds have very low effective concentration, with cohumulone having the lowest. So I focus on cohumulone's binding mechanism the most. But just overall, taking into account all the ligands, there were really 2 trends that dominated. So if the ligand bound to sp3, it formed hydrogen bonds with the residue shown in pink, and this tyrosine on the 5th helix was particularly important in binding. But in sp10, it would form hydrogen bonds shown in pink here. So these are 3 asparagenes that were commonly found in hydrogen bonds. But in the nonpolar residues are shown in blue. And the biggest difference between the nonpolar residues is that the phenylalanine on the 5th helix was also really important for nonpolar interactions in ligands that bound to sp10. And for example, you can see the 2 different binding poses for cohumulone in sp3 and sp10. And just looking at it at a glance, you can see that these binding poses are pretty different. And the energy that we calculated, so the unified cavity energy and the snap binding energy, like the sp3 had a higher unified cavity energy, but a lower snap binding energy, while sp10 had a lower unified cavity energy, but a higher snap binding energy. So the energy couldn't really tell us which of these binding poses was the clear winner. So we used molecular dynamics to tell us a little bit more about the binding poses. And so for those of you who are unfamiliar, molecular dynamics is a computer simulation method for analyzing the movement of molecules over time. And so my procedure was, first I inserted the full protein into a membrane and solvated the system with water. And then I slowly relaxed the protein in the system by first fixing the membrane in the protein and just allowing the water to move around. And I allowed the system to equilibrate for 500 picoseconds. Then I just fixed the protein backbone and allowed everything else to move around, and the system equilibrated for 5 nanoseconds. And then no atoms were fixed, and the system was allowed to equilibrate for 10 nanoseconds. And these graphs show the RMSD value, or root mean square deviation, of the protein throughout the trajectory. So RMSD is a measure of how much the protein is moving around. So you want to see a lower RMSD value that does not fluctuate over time, because this signals a stable structure that's maintaining its conformation throughout the entire simulation. And so I performed molecular dynamics on both sp3 and sp10. And in the graph, the black is the RMSD value of the full protein, and in red is just the 7 helices that go through the membrane. And as you can see, the RMSD of the sp10 complex was much lower and more stable throughout the trajectory, which is an indication that the sp10 complex is probably more stable. And also, but in both cases, the full protein RMSD value was a lot higher. And this is likely because the full protein also includes the extracellular and intracellular loops that connect all the helices. And these are a lot more flexible and tend to move around a lot more. And so also, if we look at cohumulone's position after 1 nanosecond in pink and in 10 nanoseconds in blue, you can see that the binding pose fluctuates a lot more in sp3. Just like looking at it, these two poses are very different. And this is an indication that this is not a really stable binding pose, and that important contacts are getting lost throughout the trajectory. But in sp10, you can see that even after 10 nanoseconds, the binding pose is pretty similar. And this is an indication that the binding pose is stable and that important contacts are being maintained throughout the entire trajectory. This is also reflected in the RMSD values of cohumulone throughout the trajectory. So you can see here in the sp3 complex, the RMSD values fluctuating a lot, and it's also higher than in sp10. And it also kind of marked where hydrogen bonds were breaking and forming, and you kind of see how that correlates with changes in the RMSD values. And you can just see, yeah, cohumulone is moving around a lot throughout the entire trajectory. But in the sp10 complex, it's staying pretty stable throughout the entire trajectory. And so these are graphs that show the interactions between cohumulone and TAS2R40 throughout the trajectory. And as you can see in the sp3 complex, the energy seems to be fluctuating around a lot, but it almost seems like the total energy is higher for the sp3 complex. But in both cases, nonpolar interactions contribute more than polar interactions. But the sp10 complex had much more favorable van der Waals interactions, which is likely what's leading to its stability. And so from the molecular dynamics study, we can conclude that the sp10 complex is likely a better representation of cohumulone's binding pose. And so this is just a close-up of what the binding pose looks like. This is the final binding pose of cohumulone to TAS2R40 after the 10 nanoseconds of molecular dynamics at 310 kelvins. And cohumulone is making hydrogen bonds with these 3 asparaginines in pink. And these contacts are maintained throughout the entire trajectory, and they stay between 2 to 4 angstroms throughout the trajectory. And then in blue, you can see the nonpolar residues that are stabilizing the alkyl chains of cohumulone. And so now that we have a structure of TAS2R40 and a pretty good understanding of its binding mechanism, the next steps of my project are to modify the ligand to optimize binding. And we would also like to design a ligand that selectively activates TAS2R40 and a ligand that selectively inhibits TAS2R40. And we'd also like to experimentally validate results with collaborators at the University of Arizona and the University of South Florida. And hopefully all of these results can just be used to enable investigation in TAS2R40's role in hypogeusia and also inflammatory lung disease. So I would just like to thank my mentors, Bill Goddard and Soo-Kyung Kim, for all of their guidance this summer. I learned so much from them, and they were so patient with me and helped me accomplish a lot this summer. And I also would like to thank Andres Jaramillo Botero for all his help with my computer difficulties, and also the SFP office, Candice, Maria, and Carol, for making my summer at Caltech possible. Thank you for making this the best summer that it could be. And thank you so much for listening to my presentation. I look forward to hearing your questions during the Q&A session. Thank you.